Alright guys, welcome back to Underground Science. In this video we're going to be talking about non-homologous end joining. Okay, so this is relating to double-stranded DNA repair. Okay, so if you have a double-stranded break in your DNA, then this is one of the ways you can repair it, which is non-homologous end joining. Let's talk more about this. So it says the name is non-homologous end joining, right? So that means you don't need a homologous template like you would in homologous recombination double-stranded DNA repair, right? So basically the main thing we have to know here is not like each and every protein that's involved in everything. The main thing to understand is that you get a double-stranded DNA break, right? So this portion's gone. Then you just recruit enzymes and you rebuild it back. But the only problem with non-homologous end joining or non-homologous end joining double-stranded DNA repair is that this isn't accurate at all, okay? So what I mean by that, it's not accurate because it's basically prone to making a lot of mistakes, this non-homologous end joining method. Alright, so it's non-homologous end joining double-stranded DNA repair isn't that accurate. Okay, it's not that accurate. This process is prone to, making mistakes. So what mistakes are we talking about? So we're talking about certain insertions, deletions, right? So like in this process you may add like too many nucleotides, or you may delete too many nucleotides in the process of repairing the double-stranded DNA break, or you may see some translocations that occur, right? So this isn't accurate, right? Like if you compared this to homologous recombination DNA repair, which is for also double-stranded DNA repair, like that was accurate. You did not lose any DNA. This one, it's the opposite. You probably will lose DNA. It's not accurate. This process is prone to making mistakes. All right, so that's the main part about non-homologous end joining. Okay, you don't need a homologous template. You make a break, recruit enzymes, rebuild it, and just know that's not accurate. It's prone to making a lot of mistakes like translocations, insertions, and deletions. All right, so that's basically it for this video. It's a short video because it was just understanding non-homologous end joining. That's it. Now we can understand ataxia telangiectasia, which is an important disease to know relating to non-homologous end joining. Alright, so see you guys in my next video. Thanks for watching.